Okay, this is chapter 2. And this gives us some overview of some of the big pictures in microbiology, um, how we name things, how we classify them, and some things about microscopes. Okay, so after we finish with this chapter, you should be able to explain how microbes are named, and this is a very important point. We can illustrate how organisms are classified into the tree of life, distinguish between the different forms of microscopy, and finally being able to construct and label a typical eukaryotic and prokaryotic cell. One of the first people who classified organisms was Carlos Linnaeus, and he was a botanist. And he is responsible for discovering hundreds of new animal and plant species. And he came up with a naming system to use the binomial nomenclature system. Okay, so binomial means two names. Okay, so what's in the name of a microbe? So all the microbes generally have a binomial official scientific name and their common name. So, like I said, binomial means two names, and the first word is the genus, and the second word is the species. Okay, so you're always going to have two words by having a genus and a species. And one of the sort of best known examples, since almost everyone knows what Escherichia coli is. Now, of course, a lot of people will just say E. coli, but the official word is Escherichia, so Escherichia coli. Okay. So the first word, Escherichia, is the genus name, and coli is the species name. In this case, there are very few different Escherichiae. Is sort of different from something like Streptococcus, where there are a number of different Streptococci bacteria. There are a number of different things in that genus. And so E. coli or Escherichia coli is one specific microbe of that genus and species. Okay, like I said, we're going to have binomial or common names, and common names. And the binomial name is going to be the strain or the subspecies. So, for example, one of the things you hear about in terms of meat preservation is Escherichia coli 0157H7. Okay, so that's a very specific type of E. coli, and that type is of the O antigen 157 and H antigen number 7. Okay, so when you're looking at information about outbreaks, you're often going to see Escherichia coli 0157H7. Another common bacteria is Salmonella enterica typhi. Okay, now this is a causative agent of typhus. So the official name is the genus is Salmonella, the species is enterica, and the subtype is typhi. Okay. Sometimes people are only going to use the common name So while the official name for this bacteria here is Streptococcus pneumoniae, normally known as pneumoniae, sometimes this might just be known as pneumococcus. Okay. Here's another good example. Neisseria meningitis, meningeal cocci, the causative agent of meningitis. And again, the common name sometimes is a little bit easier to get across the point, but when speaking scientifically, you want to use the entire genus and species name. Okay, how we catalog the different forms of life is taxonomy. And in general, most things live up to this higher order for different groups. And I'm sure you've probably heard this before: kingdom, phylum, class, order, family, genus, species. Okay, and you can add subspecies there too. Now, to try to give you a sort of overview of what this actually means, the kingdom could be something like department stores. Okay, so that's the big picture of what's going on. The phylum might be the watch counter. Okay, the classes, we could have men's watches, which would all be in a department store. We could have wristwatches. Then the family would be dress watches. Genus could be the brand, the fossil brand, and then the subspecies would be a fossil red, which would be a very specific watch, and you could also have one that would have the serial number on it. Okay, so this is how you'd classify this watch, this happens with all organisms. So there is one domain or one thing that's higher than a kingdom, and that's the domain. Okay, so we can have eukaryotes and prokaryotes. If you're thinking about a human, you would have something that's a eukaryote, that's an animal, that's a chordate, which means it has a backbone. Humans are mammals. Humans are also primates. They're hominids, and Homo sapiens is the genus and the species. Now, in the microbial world, we could have a eukaryote of something like yeast, where again we're in the domain of eukarya, then fungi, then actomyces, Saccharomyces, and Saccharomyces cerevisiae is brewer's yeast. Then in bacteria, we have the group of, or the phylum of protobacteria, gamma protobacteria, the order of Enterobacteriae, the family Enterobacteriae, then we have the genus, and then coli would be the species. Okay. And so here's a representation of the 5 kingdoms. Your animals, plants, fungi, and Monera are down here. I think we all understand what animals and plants are. Where it gets a little more funny is in the molds, in the fungi. We can have a number of different fungi, everything from something like a mold to a mushroom. And then in the Monera class, you have all the different kinds of bacteria and algae and things like that. Okay. And remember that in this course, since we're mostly working with microbes, we're mostly going to be in the Monera class looking at bacteria. One thing that you won't see on either of these lists, actually, in the list I'll show you in a minute, is viruses. Okay, viruses are not living. Okay, viruses are just pure bits of organic material. Okay, so they're not living. Okay, now Carl Woese actually developed this 3-domain system, and this is kind of the overriding thing nowadays. The 5 species or the 5 kingdom theory has kind of gone out of play, and so we now divide things into domains. And one of the things I think is so great about Woose's classification is that these are based on molecular properties of these things. Okay, and so we can divide these into 3 broad categories of bacteria, archaea, and eukarya. We sort of have a feel already for what bacteria are. Archaea are bacteria-like things, but things that live in extreme environments. And then eukarya are typically the big things like animals, plants, and fungi. Those are mostly multi-organ or multicellular, but there are a number of single-cell eukarya also. Okay. And like I said, the modern system uses this 3-domain system. Okay, and this is called the tree of life because if you look at the very start of this tree, okay, the one universal ancestor is at the base of the tree, and then everything goes off of that tree, branches off from there. Okay, so there was some original cell, and that divided, and then was able to divide to make all the bacteria and the eukarya, and then at some point after things had split into the eukarya, the archaea domain, those things were there in that middle domain. One of the real important things about bacteria is that these are cells with no nucleus. Okay, they have very, very little internal structure, and so prokaryote, karyote means nugget, so pro-nugget means without a kernel, okay, and those are the ones that don't have a nucleus. Our archaea also don't have a nucleus, okay. It's only when you get into the eukarya do we get a nucleus. Okay, so that's kind of the surest way to be able to tell these different kinds of cells apart by whether or not they have a nucleus or not. Okay. You might remember that I introduced in the last lecture that a microbe was anything that can't be seen without the use of a microscope. Okay, and so there's been a very close relationship between the development of new and better microscopes and discovering more and more microbes. Okay, so in general, when we are talking about the microbial system, we're looking at micrometers. Okay, and I know that it's spelled micrometers, but it's pronounced micrometers, and there are 1,000 micrometers in a centimeter. Okay, and so those are where the majority of our microbes, our bacteria, are being placed. Now, when you get to an even smaller structure, then you can use nanometers to look at viruses, and these viruses are too small to be seen with a light microscope. So for a long time, people didn't think there were viruses because they couldn't see them under the available microscopes. So here's a classification of the different types of organisms and what kind of microscope can be used to see them. Okay, so for things like molds or tapeworms, you can use the unaided eye, and these are maybe on the order of a centimeter or so. Things that are very small, like the bacteria or some of the fungi or yeasts, those can be seen with a light microscope. Okay, and then those things that can only be seen with an electron microscope, like typical viruses or sometimes some molecules themselves, you're going to need an electron microscope to be able to see those. So your typical light microscope, generally called a compound light microscope, has a few key features, and the magnification is all in these eyepieces here. Okay, and you can see that in this close-up here. You have one of the lenses here, and then your sample would be right there. Okay. You're always going to have a condenser, which will help focus the light. And to see very small things, you need something called oil immersion, because when the light is going through your sample, this space can diffract the light. So if you put a drop of oil on the slide there, then you get sort of a constant layer between the objective lens and the sample, and you get much less distortion. Okay. Now there are a couple other types of light microscopy, and so again, these are all dependent just on light. In dark field microscopy, you're actually putting a filter on so that the background is completely black and the cells you want to see are white. Okay. Now this actually shows the most prominent example of dark field microscopy when it's used to look for spiral-shaped bacteria. In fluorescent microscopy, we add some chemical to the cells that will only glow under fluorescent light, and we can then gather that type of light through a bunch of filters to be able to get pretty pictures. This can be used to do some diagnostics. You can maybe add a fluorescent compound that will only light up a certain disease, and you can see if you have that in your sample, then your samples will all glow. Okay. Now in electron microscopy, again, very late to be discovered, and the sort of big advantage here is that you don't have to look at the object. You're actually bouncing electrons off of the object to be able to see it. Okay, and there are two types of electron microscopes: the transmission electron microscope, the TEM, and which that can get to about 200,000 times the size of the actual object. And then a scanning electron microscope, an SEM, is about 20,000 times. Okay. Now, I kind of like this slide. So just to show you how different types of microscopes can give you different insights. If we look at some Pseudomonas, here we're looking at a standard light microscopy and seeing all the very, very small cells in this object, in this slide. Okay. Now if we're in a TEM, okay, then we could actually see individual cells, and you know, sometimes in this case these cells are dividing. And here in an SEM, we're seeing the outside of the cells. Okay, so an advantage of a TEM is you're seeing the inside of the object, whereas in an SEM, you're seeing the outside of the object. Okay, so now we're going to compare some of the internal structures of eukaryotes and prokaryotes. Okay, so here's a typical eukaryotic cell, and again, the big thing that makes this a eukaryote is that it has a nucleus. Okay, and your DNA is contained within the nucleus. Now, you have a number of different membranes. So, for example, the nuclear membrane around the nucleus. You can have endoplasmic reticulum, okay, Golgi apparatus, endoplasmic reticulum, Golgi apparatus. You can also have membranes for things like the mitochondria, and you have a number of different vesicles inside of these cells. Okay. And every cell type is going to have some ribosomes, and because ribosomes are the place where proteins are made. To generate energy inside of a cell, you need a mitochondria. Okay. Now in a prokaryotic cell, we have no nucleus, and the DNA is basically free floating in the center. Okay, you have a cell wall, and sometimes an outer shield on the cell in general, but there are no internal membranes. You have the ribosomes still, okay, but there are no mitochondria, there are no endoplasmic reticulum, there's no Golgi apparatus, none of that inside of a prokaryotic cell. Okay, so what we wanted to learn here is that our microbial world is fairly diverse and is going to have a binomial nomenclature system. Things all originated from an original cell type, and that is in the tree of life. There are two of the three domains that are prokaryote, the bacteria and the archaea. Okay. And here's something for you to think about, and maybe a thought question we might put on some sort of an exam. But are prokaryotic organisms simpler than eukaryotic ones? Okay. And that's a really complex question, because one can say that, well, they're simpler in terms of not having more organelles, but yet a bacterial cell can do everything, if not more, than a eukaryotic cell. So is that, do they make up the simplicity some other way?